Good morning, everyone. I'm delighted to welcome you today. Today, we'll be exploring a very important topic, the parametric approach to batch release in sterile manufacturing. Our focus will also include the key global regulatory expectations from agencies like the FDA, EMA, and PIC-S. Over the course of this presentation, I'll guide you through the principles, challenges, and regulatory perspectives related to this approach. So, let's get started and dive into this fascinating subject. Parametric release, or PR, is a science and risk-based decision model that allows us to confirm batch sterility assurance differently from traditional methods. Instead of relying solely on the standard sterility test, PR demonstrates that sterilization parameters have been properly validated and achieved the necessary microbial kill. This approach focuses on directly measuring critical process parameters like temperature, pressure, the FO value, and exposure time. By doing so, we can confirm that the sterilization conditions have effectively achieved microbial lethality without waiting for the end product test results. Importantly, this method aligns with modern quality frameworks such as ICH Q9 for quality risk management and ICH Q10 for pharmaceutical quality systems. It supports the shift from end product testing to a process-based assurance approach, ultimately enhancing our confidence in product sterility and quality throughout manufacturing. Traditional sterility testing has some inherent limitations that we should be aware of. For starters, it's probability-based, which means it only detects a fraction of contaminated units, so there's always a chance some contamination could be missed. It's also destructive, meaning that the samples we take can't be returned to the batch, potentially wasting product and resources. Plus, it takes a long time, up to 14 days, to get results, which can delay product release and impact supply chain efficiency. Another challenge is the risk of false positives or negatives, which can lead to unnecessary batch rejection or, worse, releasing a contaminated product. That's where parametric release comes in as a game changer. It offers real-time, scientific confirmation of sterilization lethality, so we don't have to wait weeks to be sure. This allows for faster batch release, ultimately improving supply chain velocity. Parametric release also provides higher sterility assurance because it's based on validated sterilization cycles, giving us confidence in the process rather than just testing the final product. It reduces our dependence on end product sterility tests, which are limited and time-consuming. Additionally, it helps us gain better process understanding and life cycle control, so we can optimize and monitor sterilization more effectively. Globally, PR is recognized as a superior alternative, especially for terminally sterilized products. So, why choose parametric release? Because it addresses the limitations of traditional methods, accelerates production timelines, and enhances overall sterilization confidence. Now, let's discuss the scope of parametric release and where it applies. Primarily, it is applicable to terminally sterilized products. These are products that undergo a final sterilization process that ensures they are sterile before leaving the manufacturing process. The main methods here are moist heat sterilization, such as using steam or autoclaves, which are very common in the industry. Additionally, some radiation-sterilized products may also qualify for parametric release, but this is on a case-by-case basis. It requires scientific justification to ensure that the sterilization process effectively guarantees sterility without the need for traditional biological indicators. Now, it's important to note where parametric release does not apply. It is not suitable for aseptically manufactured products. These products are produced in a sterile environment but do not go through a terminal sterilization step, so we can't rely solely on process parameters to confirm sterility. Understanding the scope helps us determine when parametric release can be confidently used, streamlining release procedures while maintaining safety and quality. Now, let's take a closer look at the key regulatory bodies and their perspectives. These organizations play a crucial role in shaping the landscape we're discussing today. They set the standards, enforce compliance, and influence how the industry evolves. Understanding their viewpoints helps us anticipate future changes and align our strategies accordingly. As we explore each one, keep in mind how their priorities and regulations impact not just compliance, but also innovation and growth within the sector. This connection underscores the importance of engaging with these bodies proactively to stay ahead in this dynamic environment. Let's take a closer look at the regulatory basis for sterilization processes. It's grounded in 21 CFR Part 211, which sets the standards for drug manufacturing, along with FDA sterility standards and current good manufacturing practices, or CGMP. These regulations ensure that sterilization methods are both safe and effective. Moving on to the FDA's key requirements, a validated sterilization cycle is essential. This means we need to demonstrate lethality, typically a FO of 12 or greater for moist heat sterilization, to confirm that the process reliably kills all microorganisms. It's also important to provide evidence of consistent heat penetration and load reproducibility, so we know the process works uniformly every time. Robust CPP monitoring is another critical aspect. Continuous measurement of temperature, pressure, and FO, as well as load mapping, helps us stay on top of process control. This real-time data allows us to quickly identify any deviations and make adjustments as needed. Real-time data integrity compliance is equally vital. Using 21 CFR Part 11, compliance systems ensures that electronic records, audit trails, and traceability are maintained properly, fostering trust and accountability in our data. A comprehensive quality risk management, or QRM program, is also part of the picture. Based on ICH Q9 guidelines, it involves risk assessments that help us identify potential issues early. Scientific justification is key when it comes to eliminating sterility tests without compromising safety. Finally, the regulatory approval pathway involves submitting a supplemental application. This includes validation data, CPP definitions, and a lifecycle management program to demonstrate ongoing control and compliance. Overall, the FDA expects that we not only meet these technical and regulatory standards, but also integrate them into a cohesive, well-documented process that ensures product safety and quality throughout its life cycle. Let's take a closer look at the guidance basis outlined here. As you can see, it's rooted in EU GMP, specifically Annex 1 from the 2023 revision, and also references Annex 17. The EMA emphasizes that parametric release is now considered the preferred alternative for terminally sterilized products. This approach relies on demonstrating consistent lethality and uniform heat penetration during sterilization cycles, rather than traditional sterility testing. A critical aspect is real-time monitoring of all key parameters, which ensures that every cycle maintains the necessary control and consistency. Additionally, the approved quality risk management strategy must include rationales for eliminating sterility testing, making sure that all decisions are thoroughly justified and documented. There are also extensive requirements related to load mapping, sensor placement, and periodic requalification. These are essential to validate and maintain the sterilization process over time. Importantly, any parametric release program should be integrated into the pharmaceutical quality system, aligning with ICH Q10 principles. Overall, the EMA's expectations revolve around ensuring a robust, scientifically justified approach that emphasizes process control and continuous validation, aligning with the broader goal of maintaining product quality and patient safety. Let's look at the guidance basis here, which is PI 12 and its Annex 1 alignment. Essentially, this means our practices are aligned with the PIC-S requirements, which are harmonized with EU GMP standards. This alignment is important because it supports the overall process robustness and compliance. PIC-S requirements are quite comprehensive. They strongly emphasize the importance of digital trails that are inspector verifiable, meaning all electronic records must be reliable and traceable during inspections. Consistency over time is also crucial, so cycle reproducibility is emphasized. This ensures processes can be reliably repeated, maintaining quality and compliance. Another key point is the qualification of autoclaves, which must include redundant monitoring systems. This redundancy helps ensure sterilization processes are consistently effective, even if one monitoring component fails. And of course, the training and competency of operators are fundamental. They need to be well trained to handle equipment properly and understand the criticality of their roles. Prior licensing is also a requirement, and it involves submitting a detailed process and product registration dossier. This step confirms that all parameters are well documented and meet regulatory expectations before operations begin. Overall, PICS expectations reinforce a strong, structured approach to manufacturing quality, aligning closely with EU GMP, and emphasizing the importance of verifiable, consistent, and well-monitored processes. This forms the backbone of a compliant and reliable manufacturing environment. On this slide, we're looking at the critical expectations across major regulatory agencies like the FDA, EMA, PICS, and WHO. These standards are essential to ensure the safety and efficacy of sterilization processes globally. First, let's talk about process and validation. It's vital to have a scientifically justified sterilization process, meaning it's based on solid research and understanding. Plus, we need validated sterilization parameters, often called FO, which confirms that the process reliably achieves sterilization. Identifying clear critical process parameters, or CPPs, is also fundamental. These are the key factors that influence the sterilization outcome. Demonstrating that heat or energy is distributed evenly throughout the load is crucial to avoid any cold spots or underprocessed areas. Control of routine load configurations ensures consistency from batch to batch, maintaining process reliability over time. Moving on to data integrity and digital controls. These are about ensuring that all sterilization data is accurate, complete, and unaltered. Systems like SCADA or PLC, which are compliant with standards like Part 11 or Annex 11, help us manage this digital data securely. Applying ALCOA Plus principles to all sterilization data means our records are attributable, legible, contemporaneous, original, accurate, complete, consistent, and enduring. An audit trail review becomes an essential part of batch disposition, allowing us to verify all process steps and data are properly documented and reviewed. Now, let's discuss lifecycle management. This involves periodic requalification of sterilization processes and equipment to confirm ongoing performance. Conducting annual product reviews and trending CPP performance helps us spot any deviations early and maintain process control over time. Finally, these are common global requirements, meaning they're recognized and enforced across different regions. So aligning with these expectations is key to ensuring our sterilization practices meet international standards and safeguard public health. When it comes to a robust documentation package, there are several key components that we need to include. First, core documentation is essential, which covers sterilization validation protocols and reports, cycle development studies, CPP justification documents, real-time cycle monitoring records, electronic batch records, and audit trail review reports. These elements ensure that every step of the process is well documented and traceable. Next, we have to consider deviation and change control. Regulators expect us to have predefined decision trees for addressing CPP deviations, which helps streamline decision-making during unexpected events. It's also important to classify deviations clearly as critical or non-critical, so we know how to prioritize our responses. When deviations occur, thorough investigations are vital. These investigations should be supported by trend data, historical cycle performance, and evidence of heat distribution. If a CPP failure happens, requalification becomes necessary to confirm that the process still meets all required standards. Finally, the documentation requirements are not just about record-keeping. They're about demonstrating compliance and ensuring process integrity. All these elements together form the foundation of a solid documentation package that supports quality, compliance, and continuous improvement. Regulators are increasingly expecting us to modernize our processes in line with Pharma 4 standards. One key area is the implementation of automated sterilization systems that include real-time flow of calculation capabilities. This means we can monitor sterilization processes as they happen, ensuring precision and compliance. Another important aspect is the integration of electronic batch records, or EBR, with SCADA and PLC systems. This integration helps us streamline data collection, improve traceability, and enhance overall process control. Cybersecurity controls are also crucial, especially for sterilization equipment connected to digital networks. Protecting these systems from cyber threats is essential to maintain integrity and compliance. We're also seeing the value of predictive analytics, which can forecast cycle performance and detect anomalies before they become issues. This proactive approach helps us maintain quality and efficiency. Additionally, advanced sensor technologies and IIoT-enabled monitoring provide real-time insights into equipment performance and environmental conditions. This digitalization aligns perfectly with Pharma 4 expectations, enabling smarter, more responsive manufacturing. Overall, these innovations aren't just about technology. They're about transforming our processes to meet regulatory expectations, improve quality, and stay competitive in this evolving landscape. Now, let's look at some of the key benefits this approach offers to manufacturers. First, we see a significantly faster batch release process. Instead of taking up to 14 days, releases can happen within just a few hours, which really accelerates the overall production timeline. Next, there's the higher sterility assurance level, or SAL, which means we can be more confident that our products are truly sterile. This directly enhances safety and quality. We also observe a reduction in sterility test failures and false positives. This not only saves time, but also reduces unnecessary delays and rework, improving efficiency. Lower inventory and warehouse holding times are another benefit, which helps reduce costs tied up in stock and storage. Additionally, this method improves regulatory confidence because it's grounded in scientific rigor. Regulatory bodies tend to trust processes that are transparent and scientifically validated. All these improvements lead to reduced manufacturing cycle times and better cost efficiency, ultimately making the entire process more streamlined and economical. So overall, these benefits not only enhance product quality and safety, but also provide a competitive edge by making manufacturing faster, more reliable, and more cost-effective. Let's start by emphasizing that a structured approach is essential to ensure compliance throughout the process. First, we conduct a feasibility study to assess product suitability and equipment capability, which helps us identify potential challenges early on. Next, we focus on enhancing sterilization validation. This involves mapping out the process, developing effective cycles, and performing faux demonstrations to confirm effectiveness. Moving on, the QRM and lifecycle program are critical components. We utilize tools like FMEA and hazard analysis to proactively identify risks and determine critical process parameters, ensuring we manage issues before they arise. Integration of digital systems, such as PLC and SCADA upgrades, is also vital, especially to meet Part 11 compliance requirements and enable real-time monitoring and control. Updating SOPs and redesigning EBR workflows help maintain a clear audit trail and streamline batch disposition processes, which are crucial for compliance and quality assurance. We also need to perform stability and comparability studies to demonstrate product consistency over time and across different batches. All these efforts support our regulatory submission and approval process. Once we validate the process through pilot batches, we can move toward full implementation. This is where our implementation roadmap becomes essential, guiding us step by step to ensure a smooth transition and sustained compliance. Parametric release, represents a significant scientific and regulatory advancement in how we manufacture sterile products. By utilizing validated sterilization parameters, digital controls, and rigorous QRM principles, it offers a more reliable way to ensure sterility compared to traditional sterility testing. This approach allows us to confirm product safety based on process control, rather than waiting for test results, which can speed up release times and improve efficiency. Global regulators are increasingly expecting us to adopt parametric release as a strategic, science-driven, compliant method. This shift supports faster product release, enhances overall quality, and strengthens lifecycle management. Ultimately, embracing parametric release aligns us with evolving regulatory expectations and demonstrates our commitment to innovation and excellence in sterile manufacturing. For further support, please contact us. We're here to help with any questions or additional assistance you might need after this presentation. Thank you so much for your time and attention today. Remember, we're always available to guide you through any next steps or provide more detailed information. Your engagement is truly appreciated, and we look forward to working with you further.